and I'm just going to be lecturing in a subdued way for the rest of the semester. So here we are, and welcome back, and we're rolling in chapter 18, and it's a very important chapter for us in our course, and it's about the cell cycle, and so we're in kind of the heart of that, and we want to keep following up on exam 1. So, I took time to write that NSKE video that explains my grading, and really have spent a lot of time in my life thinking about grading, and critical a lot of different kinds of grading systems that instructors use. And so you should be very comfortable and confident to understand how you got the grade you got on your exam and how that helps you understand where you stand in the course. And then I talked about the answers, and we want to still understand those, and we want to keep following up on that. And so we can meet. I think I've only met with one student following up on questions from the exam, stuff like that, maybe two. But we can continue to do that because we're trying to keep learning. So that's a big part that we're still dealing with. And then Riley's still sick, so he's going to come as soon as he can, as he's healthy again, and tell us about the third visit of CAR T cell therapy, which will be about cell response. And one thing that CAR T cells do is they go through the cell cycle. They proliferate some, but they also make other types of cell cycle responses in order to play their role in the immune system. So this week you should be wrapped up reading chapter 18 and go on to chapter 19, which is about programmed cell death and stem cells. Okay, so that's where we're at, and that's what the chapters look like in this section of the textbook, and they're all focused in our author's entire career, focused on cancer. Okay, and so where we're at is we're talking about START, the G1S transition, the restriction point, and that's where the major cell cycle regulation occurs, like for things like PDGF under a SCAP. And we talked about that, but you just really have to know that in and out, up and down. And it fits in here at the end of G1 in the cell cycle. Historically, it just looked like nothing special was happening at that time. They called it interphase. You can't really see any of it under a microscope. But biochemically and genetically, scientists determine all the factors, and most significantly, the factors that regulate the cell cycle are really almost all orthologs in every single eukaryote. So the work again was originally done in yeast. The yeast caused bread to rise. Okay, so just to look a little bit more at the phases of the cell cycle, I just want to add a couple of words here, and that is that we start in G1, and when the cells are arrested at G1, they really typically have the whole genome, and then they're going to replicate that. Now, in almost all of our cells, we actually have two genomes when the cells are at start, because we inherited one genome from mom, it was in the egg cell, and one genome from dad, it was in the sperm cell. And so I'm just going to slide over here and say that a hallmark feature of G1 is that that's where start is going to be, the major regulation point. And during G1, the cells are said to be diploid. And that means they have two genomes, one from mom and one from dad. In humans, there are, in one genome, there are 23 chromosomes. And the chromosomes are named just by numbering them from longest to shortest. So the longest chromosome is just chromosome number 1, and on down the list. And we'll just look at one of the chromosomes. Let's just say we look at chromosome 3. And at G1, really typically, you have one chromosome 3 from, say, your mom, and one from your dad. And that's really typical of animals in general. Okay? And so they're diploid. And they each, chromosomes, they have the same genes. Let's just say that's the insulin gene. I don't know what genes are in chromosome 1, but let's just say the insulin gene. And so you got a copy of insulin from your mom and one from your dad. And these chromosomes are really similar. They have the same genes, but they might have different versions of the genes. So you might have gotten a different allele of insulin from mom and one from dad. So an allele is simply a version of a gene, and that could be one allele, and that could be a different allele, either homozygous or heterozygous type of genetic talk, right? Like that, okay? And so those chromosomes are really similar, but they're not the same, and they're said to be homologous. So we're just going through some terms, sort of bookkeeping terms here. So in G1, the cells are diploid because you have homologous chromosomes. You have 2 genomes and 2 chromosomes for each of the 23 chromosomes in humans. And the homologous chromosomes are referred to as homologs. And that just means they're these similar chromosomes like that. So that's where we're at on that. Now, when we go through S phase, we're going to duplicate all those chromosomes, double the total number of chromosomes, and then we'll be in G2. So let's just look at G2 and see what's happening in G2. And I think I'll best just to erase this and write in G2, those chromosomes have been replicated, and you would draw them like this. And that could still be chromosomes 3, but you could see we doubled them. And now the cells are said to be tetraploid. And we still have the homologous pair. The homologous pair has been duplicated. And so when we have duplicated chromosomes, these are identical, like photocopies of each other, because that's what S phase does. It makes an exact duplicate. And these are said to be sister chromatids, and that's our last term. And over here, these would also be sister chromatids. So hallmark feature of G2 is we see sister chromatids. They're not in G1, they get synthesized during S phase. And in G2, the cells are really typically tetraploid. And when we get to the end of G2, we go through M phase, the chromosomes separate, and who separates is the sister chromatids, right? So each new daughter cell will get one of those photocopies. That's what happens during M phase, is the chromosomes separate, and specifically the sister chromatids separate. And in M phase, we divide the cell. We go from tetraploid back to diploid in G1 again.
Are you
that?
saying M phase like metaphase?
Uh-huh. Yeah, so M phase is synonymous. Well, no. Yeah, metaphase is a phase within M phase.
Okay, so
Yeah.
mitosis phase.
So, sorry, so I was just spacing out. M stands for mitosis. And so that M phase is the process of organizing the chromosomes and separating. And that's mitosis or M phase. And within mitosis, metaphase occurs. Yep. Okay, so just some nomenclature in there, just to add to that. And then we can see this. And what's going on here, let me just describe. It's going to be an instrument that's measuring cells in the cell cycle. And so let's just add some dots here. And so here's start. And if we just looked at normal skin cells, most of them are at start. So each of these dots is a skin cell like that, just in your arm. If you looked under a microscope and you looked at a patch of your skin cells under a microscope, most of them would be waiting at start. Then randomly, one after another gets a green light, one at a time, just randomly. And so in a patch of cells, there would be a few cells that would be in S phase, and a few here, and a few here, and a few in mitosis, a few here, like that. Just scattered around there, like that, because they got to go, and they got to go randomly, so a small number of them are going around the cell cycle. If we looked at under a scab, there would still be most of the cells parked at start, but there would be more cells going through the cell cycle because they've been stimulated. The green light, the light is turning green more frequently, and so that's what start looks like, is you have a population of cells, and they're mostly sitting there, and depending on the cell type, maybe a few have gone through and are just scattered through the cell cycle, and then when you stimulate start, you just turn the light more frequently green for the individual cells, and so you just see a higher percentage of cells scattered through the cell cycle. So what you can do, again, under a microscope, is you could look at a patch of normal cells, and you could see some in mitosis, because that's when you can see the chromosomes. You can either see the chromosomes here, or the rest of the time the cells look really the same and are referred to being as in interphase. And so it's really common to do that, to take some cells, look at a population of the cells, and count the cells that are in M phase. And maybe there would be 3 out of 100. So 100 cells in this view under a microscope, and just randomly 3 of them are in mitosis. And you could see the condensed chromosome, then you say they're in mitosis. And so 3% in that sample would be in mitosis, and that's referred to as the mitotic index. And it's an indirect indicator of how often the light's turning green. And scientists have been able to measure the mitotic index for 100 years, right? Because they could see it under a microscope, and they didn't understand the different steps. They didn't have a clue, how the cell cycle was regulated. But they could see the mitotic index, and they could see it changing depending on the growth. In embryos that are growing fast, they have a really high mitotic index. Who else has a high mitotic index? Cancer cells. Every biopsy that's ever taken has the mitotic index measured by a pathologist, which under the microscope counts the cells in mitosis, calculates the percent in mitosis. That's the mitotic index. And so like if a person goes to the doctor and says, I have a lump, a bump, a odd mole, like that, they say, well, let's just take some of those cells. We'll see what's going on, right? So they do a little biopsy, remove some of the cells, and then a pathologist looks at the cells under a microscope, and we know now, more and more, they're measuring different things. If it's breast cancer, what do they measure? They measure ER expression, like that. But one thing they've been doing for 70 years is every time they calculate the mitotic index. And that's this thing where you come back, and the doctor says, I'm not worried about that mole. It's not growing very fast. I think it's benign, right? That language comes from that mitotic index. And they've been talking about that a long time, right? Back in the 1950s, you could go and get a bump removed, and the doctor would say, I'm pretty worried about that bump. It's growing fast. I think it might become malignant. Like that, okay? Now there are more and more other molecular tools, like tamoxifen and cetuximab and RAS, PCR tests and stuff like that, right? More and more of those. But that's old school, that's still done. Like that, just works like that. So it's important to understand how that fits into START. It's all about START, but you don't have to measure START directly. You can measure it indirectly, okay? And so that's what's being done here. On the x-axis, this instrument is measuring the amount of DNA. So you can see down here, it's got diploid and tetraploid. It's actually calculating how much the DNA glows. And then on the y-axis, it's the number of cells. And this instrument is called a flow cytometer. And really commonly used for a bunch of different experiments. And it works with cells that are in solution. So not cells that you look under a microscope, but cells like blood cells that are in liquid, not tissue. And what is done in the instrument is the instrument takes the cells in the liquid and it flows through them, through the instrument, and it looks at each one at a time. And it measures the DNA content in each one using a spectrophotometer. And so in a normal cell like that, this would be a profile of where the cells are in the cell cycle. It's the way they measure that, how the cells are mostly in G1, but then scattered through the cell cycle. And you can see that here, where most of the cells are diploid. I didn't say that. This N is measuring the number of genomes. So a 2N cell is diploid. A 4N cell is tetraploid. And the textbook has just labeled what's happening. So there's this peak here. These are the cells in G1, and most of those are arrested at the red light. Then the light turns green, and the cells go through the S phase. So there is a small percentage of cells in S phase. And then there are typically a bunch of cells in G2. And then as they go through M phase, they go from tetraploid back down to diploid. Like that. So this is just a flow cytometer that measures the sort of depiction of the cells moving through the cell cycle. And it's not measuring the mitotic index, it's just measuring that distribution. Like that, yep. If you did this for brain cells have all stopped at G1 at the start, and they're said to be in G0. So if you did this for brain cells, it would just be one peak. They're all stopped. None of them are replicating their DNA, and none are G2. They're all just stopped, permanently stopped. And so that's true of the brain. It's really typical of differentiated cells. Differentiated cells really typically leave the cell cycle and permanently arrest in G1, which is why some people call it G0. And it would just be one peak, just diploid cells, okay? All right, this just shows, the book's talking about this and just talking about the cell cycle a little bit. This just shows that really often in early embryos, especially non-mammalian embryos that are growing really fast, like that, like the little frog and salamander eggs in the ditches around here right now that are getting laid, those develop really fast. And the interpretation is that there's a lot of natural selection pressure to form a tadpole as fast as possible so it can swim away from predators, like that. And when that's done in early embryos, they grow so fast that a lot of times they just have M phase and S phase, and no G1 or no G2. So the book is just showing you a little bit more knowledge about the cell cycle and how in some embryos, not mammals, but pretty much all other animal embryos, embryos are dividing really fast. They're able to do that because mom has filled the egg, right? The egg is typically really big. Think of a chicken egg. That yellow is an egg cell. Mom has filled it with building blocks like that. So it's just ready. It just has already gotten all the nucleotides needed to go through S phase, like that. And it's got a bunch of proteins in there already to do the work, like that. And so the book puts it in here because it turns out that although we emphasize now that the people who really discovered the cell cycle regulators were people studying yeast that causes bread to rise and the yeast that's used most commonly in beer fermentation, there was sort of one group of people who were actually studying frog eggs. And they were making great progress on frog eggs because the egg had all the components in it, and somehow they were waiting. Just waiting, right, for the egg to be fertilized, like that, already. So those people were biochemists, and they contributed to these stories also. And that's why the book is in there, has it in there as partly just to kind of inform you a little bit more of the cell cycle, but also some of the history. Yep?
So in embryos, like the cell isn't growing before it divides. So is there like a pre-programmed set number of divisions that take place before it triggers a standard cell cycle, or
Yeah,
something else?
like in fruit flies, they grow really fast because they're in rotting fruit. So there's a lot of natural selection pressure, and so they go through 17 cell cycles. And there's more to say about that, but yes, they just go cranking through them. And during that time, the embryo stays the same size as the original egg, like you said. It's not growing at all. It's just kind of just quickly dividing the cell up into, or the one fertilized egg cell into many smaller and smaller cells. Yep. Yeah. Okay. So just some more information about that. Okay. So what we want to take out of that is especially this mitotic index. So I just made a little image of this to sort of show that in normal skin, when we measure the mitotic index, there's a low mitotic index, but it's not 0. The mitotic index in the brain is 0, because those cells are arrested, permanently differentiated. But in our skin, there's that constant replenishing that's going on every day, right? Our skin cells die and get rubbed off. Then, under the scab, the mitotic index is going to be higher, like that. And the mitotic index is always low, like maybe this would be like 1%, and this might be like 3%. Doesn't change a lot, but it's a significant increase. Yeah?
Yeah, you said normal skins cell for the first one?
Yeah.
And the
Yeah.
second one, is it the same normal cell but under the scab?
Yeah. Yep. With the woman that has skin cancer, high mitotic index. Doctors are worried, that thing in her skin is growing fast, and they're worried about that. Could spread and really, become malignant. Yep. All right, so here's just a thing I'll read just this one highlighted point, but here it is, they're using mitotic index in a research article, and it says mitotic index is predictive for survival of canine cutaneous mast cell tumors. So canine means dog, so we're just talking about pet dogs, and dogs and other pets get cancer. And sometimes people have that tested at the doctor. So same thing, the doctor would take a little bit of tissue from that dog's cancer and look at it under a microscope, like that, okay? So they say in the abstract, it says the median average survival time for dogs with a mitotic index less than 5 was significantly longer. Those dogs lived for about 70 months. Then for those with a mitotic index greater than 5, those really just lived for 2 months. So see, they're just using that mitotic index like that, and it's that predictive, right? Really powerful predictive tool. And what is it showing us? It's showing us start and how fast the light is turning green, right? The mitotic index is an indirect measure of something important, and it's that start. Yep.
This is a research ethics question. Did they give the dogs cancer, or did they only screen dogs that had cancer for their mitotic?
Oh, I don't know. Yeah, that's a good question. I never read this whole article. So I don't know. Yeah. There used to be a lot of dogs used in science research, and that number has gone way down. So I kind of suspect it's more just domestic dogs at a veterinary clinic that they're collecting, data, kind of like patient record data on and following them. But that's pretty dramatic, right? 2 months survival time versus 70 months survival time. And that's the kind of talk that human doctors do too, right? I don't know if I mentioned this class, but when my mom was really old, she passed away of old age several years ago, she had skin cancer on her scalp. And the doctor said, Lois, we're not even going to treat that because you're going to die of natural causes before this skin cancer kills you because it's not growing very fast. And they didn't say mitotic index, Lois, right, because she's an old lady, right? They just said, not growing very fast, not worried about it. Maybe give you some cream, but it's not it's not a chemotherapy. It's just to keep the skin from drying out. Yep. So there's a lot of that just from mitotic index. Now, here's something more. So this is something, and again, just look at it. You don't have to deal with it in great detail. here, this is a table that pathologists created to help the doctor with the prognosis. So again, the doctor takes the biopsy and then gives it to a clinician called a pathologist who's working in a lab looking at tissues under a microscope. And here the pathologists are looking at human cancers, and they're looking at a certain kind that's called GIST. And I'll just flip forward to that just to show you. It stands for gastrointestinal stromal tumor. So you don't have to remember that in detail. Just a tumor, right? We get tumors in our intestines, right? Like that. And this is one common kind, GIST. And it's one of these things when a person gets a colonoscopy, they see a polyp, right? And they want to remove those. Because polyps are kind of like a wart or a mole there that might be growing slowly, but they don't know. So let's just remove them, right? So here, what they're looking at is they remove these polyps, and they look at those under, the pathologist looks at those under a microscope, and here they're measuring the mitotic index, and then the size of the polyp. How big was that polyp, and how fast was it going through the cell cycle, right? And so that's what they're talking about, mitotic index here, and then size. And then here, these are just different positions along the length of the digestive tube, because some of the prognosis varies depending on the position along the length of the digestive tube. But this is literally what pathologists do, is they collect this data and then they give it to the doctor, because the doctor who talks to you is typically not really fancy. You know, we put a lot of faith in doctors, but they have a lot of stuff they have to understand. And then they get tables like this with big letters saying, you know, who should we follow clinically, like that, right? And so they talk about this, a gastrointestinal tumor, one of these polyps that is less than 2 centimeters in size, and a mitotic index with less than 5%, that's what it means, right? Look at 100 cells and 5% were in mitosis, could see condensed chromosome. No need to follow up. The doctor says, you know, I think you're fine, you know, let that polyp out of there, and you know, it doesn't seem like it was a problem anyway, so you're good, right? Then it says, you know, that they're, you know, very low risk. But then up here, they say more about the risk for progression, right? And you can see that as the size or the mitotic index gets higher, we go from none, very low, low, to moderate, to high. And it's a bigger polyp with a higher mitotic index. So that happens too, right? Mitotic index is a central one. And then here's an additional one in gastrointestinal tumors. Let's look at that polyp size. Where are we at in sort of the progression of the size of this growth thing, okay? Like that, so it fits in there. Now, I showed you this partly just to show you this. So that's, you know, that's old school, but still current, done all the time, right? But now they're adding more to it, right? We're learning about it in our course. So here's this Wikipedia page of the cancer we just were learning about. And now they want to start adding some molecular gene expression and mutation stories to it. So here it says, GISTs are the most common mesenchymal neoplasms in the gastrointestinal tract, right, like that. And they say what I highlighted. They're defined as tumors whose behavior is driven by mutations in KIT, 85% of the time. Later it says 95% of GISTs stain positively for KIT. Right, so now they're starting to add some molecular information to this prognosis, like that. Let's add some of this detail. And so what is KIT? And we had this on our list all the time, but this is where we talk about it in our course, just one time. And KIT is one of these 58 different receptor tyrosine kinases in our body. And we didn't talk about it for the first exam, but we talked about ERB, which we love, and PDF receptor, which we want to marry, and the insulin receptor we just know is important, right? Here's KIT. And when we think of KIT, we think, mutated in gastrointestinal tumors. On the Wikipedia page, they say that really defines it. Like they look at it and they say, what kind of tumor is this? And they say it's gastrointestinal tumor because it's got a mutation in KIT. So they really want to measure mutations in this KIT. KIT is a receptor. KIT is upstream of signal transduction pathways like RAS, which is upstream of RAS, MAC, MEC, ERK, ELK, but then ELK can activate June FOS, or ERK could also activate MYC in certain cells, right? So this is what we're talking about in the cell cycle, is KIT, receptor activities upstream of activating expression probably of cyclins to make the red light turn green. So there's mutations in it like that, okay? Look at this one too. It says, 85% out of this. It says 10% of GISTs have mutation in this, the platelet-derived growth factor receptor. And there are two forms of the gene, A and B, but there's our favorite gene right there, like that, So see, they're adding some molecular details, and they're doing that more and more with a whole bunch of different kinds of cancers, not just breast cancer and tamoxifen and ER positively, but KIT in GIST, because it's going to help inform prognosis and hopefully someday treatment. Yep.
On the previous slide, it said 85%, and then the next sentence said 95%.
Yeah, I saw that too. That's kind of a curious thing. And I think what they're saying there is that, and it also says, it doesn't really say that there, but I think what they're saying there is, even if it's not mutated, in some situations, even the normal KIT, if it's expressed, appears to be contributing to these cancers, like that, is that smaller percent. And that, we saw that right with the tamoxifen and the ER positive, right? Is that you start out and you say, ooh, yeah, KIT mutations, they're really kind of diagnostic of this type of tumor. And then you learn more and you say, of most types, 85% of this type of tumor, and maybe we're starting to see there's another different story going on. And so if we have some chemotherapeutics that would say target KIT, it would be back to that thing where you're like, okay, well, we want to know which KIT, and and if it's not KIT, like that stuff, right? Because it's like 85%, and then 10% more are KIT but not mutant, and then there's another 5% that apparently don't express KIT, like that, right? So that's, I think they're sort of starting to lean into that, that in our course KIT, but then it's going to get more complicated than that as they get more refined in understanding it. Yep.
So is KIT getting mutated in similar ways of like oncogenic related thing, or like retroviruses? How would it be getting mutated?
Yeah. So remember that. It would be getting mutated just in this human's body in their lifetime. So through some random mutation that's caused by it could be an environmental factor, or it could be our lining of our intestine. Those cells have to grow and divide. They get rubbed off too, right? So they're slowly going through the cell cycle, right, like that. So their DNA is getting replicated for our whole life, and so there could be errors in DNA replication and DNA repair, right? So it could be like an environmental factor, some kind of mutagen chemical, or it could be just that the process of growing and dividing, and then errors build up. Yeah. Yeah, that would be the idea, and definitely not retrovirus. Right? That's that 1970 thing that we kind of left behind, and these would all be modern humans with no story of retrovirus. Right? Okay? So we want to do that. We want to talk about the mitotic index, have a really good handle on that as an indirect indicator of what's happening at start, and understand what that means. How is it related to start? And then how the mitotic index is used, and then it's examples like KIT. Our story is mitotic index, polyp size, and what's going on with KIT and other genes, right? So more and more pathologists are not just looking under a microscope, but they are doing PCR assays, DNA assays, gene expression assays, looking at what's going on with these genes. And the pathologist doesn't look at every gene, but they look at the ones that have been shown to be important in gastrointestinal stromal tumors, right? So there are different ones in different tissue types. KIT probably plays a normal role at some point in this or something, like that. We don't talk about KIT in skin cancer, I don't think, because it probably never is playing a role in normal skin cells. So I just put this on here. This is our old, figure. Let's see, I got this, our old figure. Well, that would be KIT, right? We talked about it as if it was PDGF receptor, ERB. When it's the FOXO pathway, it's the insulin receptor. And here, gastrointestinal stromal tumors, that would be KIT, activating RAST and so forth, like that, downstream. Here's just another figure of that showing KIT. There's KIT, and can activate this pathway we love. And then we've said before that when we learned about receptor tyrosine kinases, we learned about 3 pathways. And here's that PI3 kinase pathway that it also activates. So just showing, what's KIT doing. And so this is interesting to see because it sort of foreshadows us going into chapter 19, like later this week and next week. So here's this pathway that's a cell cycle pathway, that's a chapter 18 pathway that's going to be turning the red light green. And it's probably going to do that by causing synthesis of cyclins. That in these cells, probably they don't have enough cyclins, and we need cyclin-dependent kinase, because when that's active, the light is green. So if we can get it synthesized, because cyclin is a protein, and it's got a gene, and if we don't have cyclin, we're going to have to synthesize it. And to activate the gene, we're going to have a transcription factor. And I don't want to start shouting, but we learned last time, and Godwin clarified, these are different enhancers upstream of cyclin, and one or the other or more could activate it. We can have MYC bind, we can have June Phos bind, we can have beta-catenin bind, we can have ERK bind, like that. And we just, we just have to be solid on that. Now, look over here. So over here, this PI3 kinase leads to activating anti-apoptosis. And so that's chapter 19, apoptosis is programmed cell death. And I said that when growth factors stimulate a cell, the cells almost always are making a response also about this programmed cell death option of whether or not they should just kill themselves. And in this case, this growth factor that binds to KIT stimulates the cell cycle and also reassures the cell, you're fine, don't kill yourself. There's this complicated interplay, and we'll kind of go through chapter 18 pretty quickly because it's complicated, because these growth factors can both stimulate programmed cell death, that maybe we should just kill this cell, and they also inhibit it in complicated networks like that. But here's our first look at that, and it's a nice visual because that's happening where? In those polyps. Yep.
What's SCF?
Yeah, so it's the ligand, the paracrine factor signaling molecule that binds to KIT. It's called stem cell factor because that's where it was first studied, but it's not a very good name for it, so that's why I don't say it, because you're like, oh, it's involved in stem cells. No, it's involved in a lot of different settings, not just a stem cell
And you
story.
said that was the ligand?
Yeah, that's what binds to KIT. But we don't need to know all those ligands, right? It's just obviously there is a ligand that activates KIT. Yeah, but it's a less important thing than these downstream stories like that and the role in gastrointestinal stromal tumor. Yes?
What does the mutation alter in KIT to cause it to be overactive?
I don't know. Yeah, what types of mutation are happening? I don't know. Yeah, we've learned that in RAS there's very specific types of mutations. In ERB there are very specific types of mutations. In MYC there are different kinds of mutations, regulatory mutations. KIT, I don't know. And could speculate, but that would be bad, right? Because I just don't know. But it would, I'm sure it's like, oh, they're very well characterized and they would fit a pattern. They would fit into some pattern of like, they're random mutations, but when you see the problem is because the mutation happened to cause trouble because it was in these kind of locations. Yep. Yep, okay. So all that is just, building up our understanding of the cell cycle, the diploid tetraploid story, and then that start. And we can measure start by going over here and looking at the mitotic index like that. Okay? So again, we looked at this, we're getting pretty familiar with this. We've learned about start. What we want to do is kind of wrap up our first little talk about start and then shift over to this one to learn a little bit more. Because it's a simpler story. A lot of stuff happening at start. And so there's start, and when, skip through this, when we look at the regulation of these, it's about regulating these CDKs. CDKs are a gene family, make a set of proteins that have different numbers. We don't really need to pay attention to the numbers, but in start, we have these three major stories. And when we talk about growth factors, the biggest story is, oh, we need to make this cyclin, because the CDK stands for cyclin-dependent kinase. You need cyclin made to activate the cyclin, CDK. So that's the one we've been talking about. We're going to go back and talk some about these binding of inhibitory proteins. Haven't really talked about them yet. And we're only going to talk a little bit about phosphorylation at start. All right? And this is just those figures that I showed last time, where the book shows, there's that RAF pathway, and we just got to get that cyclin D made, like that, cyclin made, to turn the light green. And then we had all these examples last time that, got me yelling. So ALK could activate June Phos, could activate ALK, could activate, or another pathway, beta-catenin, could activate expression of cyclin, or c-MYC, or c-MYC binding and activating cyclin, like that. So we talked about that last week and got all agitated about that because transcription factors are freaking awesome. And so now we are going to go to this other side of the cell cycle and leave start for just a little while to learn a little bit about that, because it's a simpler story and good one to learn. So we really like to talk about start. That's our number one. Now we're going to go to regulating the entering into M phase. So it's also called the G2 to M transition, and it's also called the DNA damage checkpoint. And what it's like is a washing machine where you push the button, but you forget to close the lid. And so the machine goes through some parts of the wash cycle, and then it stops. And the analogy of the lid is up because there's DNA damage. We need to try to repair the DNA and close the lid so we can go forward into mitosis. It's that simple, that there's going to be a signal from damaging DNA that's going to cause the entry into mitosis to be blocked. Okay? So the first thing to say is that the entry into mitosis is controlled by a different CDK. And you don't have to remember the numbers, but you do have to remember that it's a family, and there are different ones that regulate start and that regulate entry into mitosis. Just as I go along, I might accidentally call this one CDK1, because that's its name, and it's just, you can just remember it's CDK, but you do need to remember it's different ones, different story. And so for this CDK entry into mitosis, the story here is going to be this. There are really no inhibitory proteins that are going to play a role. So it's going to be a simpler story. Then the cyclins build up in G2. There's plenty of cyclins. That's not the regulator. The regulator is going to be these phosphates. And that's one reason we don't talk about phosphates too much at start, is because we want to go over here to the entry into M phase and talk about phosphorylation of the CDK, and they're going to be inhibitory phosphates. So when the DNA is damaged, there are going to be sites on the CDK that are directly phosphorylated and stop. Just like the lid is up. Everything's ready, cyclin's ready, CDK ready, but stop, wait. And as long as the DNA is damaged, those inhibitory phosphates are on there, and then when the DNA gets repaired, they get removed, and the cell goes into mitosis. Okay?
When you're saying that DNA is damaged, this is like not related to the phosphates, it's just the DNA is damaged.
Yeah.
because of
So
the
imagine the cell has gotten the green light, and it goes through this cell cycle. So that means it starts replicating its DNA. And one thing that can happen is that the DNA can get damaged during replication. There could be errors, and that's DNA damage. And the cell wants to fix those before the cell divides. And so it's working on fixing them, and it gets to the end of G2, and it still hasn't gotten them fixed. So these inhibitory phosphates are going to detect the presence of damaged, unrepaired DNA and say, wait. Just wait. And wait, the lid just stays up. Wait, till we get a chance to repair this. And when it gets repaired, we can remove the inhibitory phosphates. So see, it's a nice, simple story. This just shows, in chapter 18, this just shows this story. There's none of these small inhibitory phosphates. It's just a question of this CDK, again, CDK1, but this CDK, and it's a cyclin. And remember, the cyclins also come in a gene family, and they're lettered. So this is a different cyclin than at start, but it's a homologous story, right? These are paralogs, so they're paralogous proteins, and they're doing the same kind of job. This wants to kinase, it wants to phosphorylate stuff, and this wants to help activate it. Cyclin-dependent kinase. Okay, so that's what we're looking at. And this is just from an intro bio textbook, but I always really like it because it shows really beautifully a couple of things. And one is that it's this purpley thing, light purple, CDK. This CDK is always present. So it is always there. And that's interesting to see. It's got to be regulated because it's always there. And we need it to be off when? All of interphase. And we need to be on when? All of M phase. And that's what we're regulating. Because we're rolling around here through G2, CDK is present. See, and again, maybe I'll just call it CDK1, but again, you don't need to stress out about the numbers and letters. CDK1 is always present, but we want to regulate it. We just want to wait and make sure everything's repaired before we enter M phase. Then we want CDK1 to be active throughout M phase, and then we want to inactivate it. The second thing that this shows is it shows that during G2, the cyclin just gets made, and it just piles up. And so cyclin is not always present, but here in G2, there's plenty of cyclin. So that, that's not the big regulatory story here at the G2 to M transition, at this DNA damage checkpoint. CDK is always there, and now we got cyclin. Let's go, like that, right? Those aren't regulated. It's the phosphates and these inhibitory phosphates that are regulated. So that's not shown on this intro bio figure, but the first parts are shown, but we haven't seen the inhibitory phosphates yet. The last thing I want to show you is that during M phase, these are together and the protein is active, and the kinase is active all through M phase. The scientists that were studying the frog eggs were the ones who were really doing the biochemistry of this, because there was a lot of that protein in the cells, like that, right? So they purified it biochemically, and they're the people who named it, they call it MPF. And historically, back in the 1980s, that was called maturation promoting factor, because we have the frog eggs that are laid in the swamp, but they're not fertilized yet. And they get fertilized, they start to grow and divide, but they wait until they get fertilized, and so that's called maturation. And that maturation is stimulated in part by this maturation promoting factor. So it's just another name for this. And our book uses it a lot, all books do, but you don't really have to sweat that, because we know more fundamentally that CDK is always there, and it's active in M phase, throughout M phase, and it's active because it's got cyclin, and the inhibitory phosphates have been removed. Just interesting to see then at the end of M phase, the cell leaves M phase. So that would be the cell has condensed its chromosomes, so the chromosomes separate, and then the cell divides. And as it's dividing, it leaves M phase. It actually leaves M phase because the cyclins are degraded. The cyclins get broken down and it's out of M phase. And that's interesting to see because the entry into M phase is the phosphates and the exit is really the getting rid of the cyclins, like that. And that's why cyclins are called cyclins. I think I have a figure of this, is because cyclins, that cyclin that the scientists study first, goes up and down. Goes up and down, up and down, up and down. So that's why it's called cyclin. So that helps you remember cyclin, because it goes up and down. Nowadays, the guy that won a Nobel Prize for studying it, by studying the yeast that's often used in beer fermentation and working in England, he really liked riding bicycles. So nowadays he says cyclin is named because he likes riding bicycles. But it's just like, hey buddy, you're getting old. Because he really originally called it that because it goes up and down, up and down. It cycles up and down like that with the cell cycle, right, like that. So this is a really nice figure kind of from Introbio for that, the G to M phase transition, like that, get those pieces set. And so, so that's the story. We have to get to those inhibitory phosphates. When we see this, though, it's really interesting to see the CDK is active during M phase, and it's inactive during the rest of the cell cycle. And then it's really satisfying to see, what does it mean that it's active? The kinase is enzymatically active, and that means it's binding to its substrate, which are target molecules, mostly proteins, and it's adding phosphates to them. And so we want to just learn what are a couple targets of this CDK? It's active throughout M phase. Because the targets are just like, oh my God, I'm going to start freaking yelling again, guys. So the first thing is, our book shows this first thing is just a minor thing. And that is just saying that some of the targets of CDK1 in M phase there are two helper kinases. No big deal. Polo kinase and Aurora kinase. And so those get activated. They get phosphorylated, they get active. So now we got three, like the Trinity, the Holy Trinity, and they're phosphorylating targets. So CDK or Polo or Aurora, and look at the kind of stuff they phosphorylate. And we'll learn two of the targets. So what do? They phosphorylate chromatin proteins, like histones, and directly cause the chromosomes to condense, to be shorter. So remember, we've learned about histones, and they can get modified, and they can be heterochromatin, can be deacetylated, or heterochromatin, they can open up. When the cells enter M phase, they condense their chromatin even more dramatically. That's why it becomes visible. And the reason that happens is because the histones that we learned about before get phosphorylated, and they cause them to pack even tighter. And who's phosphorylating them? It's these guys. And so there's this direct definition of M phase. The chromosomes condense and become visible. And then there's another direct definition of M phase. CDK1 is active. And they just go hand in hand. It's the same thing, right? CDK1 is active, the chromosomes condense, because that's one thing it does, is it phosphorylates the histones, and the chromosomes condense. And again, it's doing it with its buddies, Polo and Aurora, because it needs a help, because there's a lot of chromosomes in there, right? And it's just going through there and just condensing everything down, and it becomes visible. What happens at the end of M phase, the chromosomes decondense and disappear. They're still there, but you can't see them anymore under a microscope. And that's because the CDK1 activity gets inactivated. How to get inactivated at the end of M phase? The cyclin gets degraded. And that shuts off this activity. It stops happening. The chromosomes have the phosphates removed from the histones and unfold again, like that, back into G1. So that's just like, it's one of those things where you're like, oh, talk about fully understanding a biochemical pathway. There it is, right? If you activate that CDK1 at any point, the chromosomes just condense. Anytime you do it, the chromosomes condense. Just, if you fiddle with it and activate it during G1 or S or G2. The chromosome is just condensed. Okay? Then the other one we're going to learn about is the nucleus in a lot of cells, human cells, and a lot of other kind of animal cells. The nucleosome breaks down. It doesn't happen that way in all organisms, but it certainly does in humans and vertebrates like that. And we'll learn about that. And that happens because there are some proteins that are like a mesh of proteins, and they're called nuclear lamins. There's a mesh of proteins that create the spherical shape of the nucleus. The nucleus itself is made of a nuclear membrane, and that has a spherical shape to it because of this mesh of lamins that are lining the inner surface of the nuclear membrane. Those also get phosphorylated by CDK1, Aurora, Polo, and they disassemble temporarily. If you know what a geodesic dome is like, it's kind of a structure or a buckyball in organic chemistry. These are kind of geometric spheres, and this lamin spontaneously polymerizes into a kind of geometric sphere. And when you phosphorylate it, it just disassembles into the individual monomers temporarily. When M phase is done, the CDK gets inactivated, the phosphates are removed, and it just spontaneously reassembles, and the nuclear membrane forms around it again, and we get the nucleus again. So just two of those, and there are more things that are happening, but those are two really satisfying ones because they're ones you see under a microscope. I see chromosomes, I see the nucleus breaking down. Why is that happening? CDK1 is active, like that. So when the cells are waiting at the G to an M transition because of DNA damage, there's no chromosomes visible, and the nucleus is intact, waiting like that, okay? So our book talks about that, and it's satisfying. Here's the figure from our book. This is a little bit more complicated, right? Not intro bio, but our figure from our book shows this too, that we get the CDKs going together, or the CDK going with the cyclin, and we get the inhibitory phosphates removed, and we're in the M phase, and then we're leaving M phase, and we're getting the cyclin degraded like that. Okay, so just a little more complicated image like that. Here we get to start seeing these phosphates. It turns out that there are both activating and inhibiting phosphates. So see these numbers here? These are the specific amino acids along the primary amino acid sequence change. So that's the threonine that's in position 161. And it's a target for phosphorylation, like that. And some of the phosphates are activating phosphates, and some are inhibitory phosphates, like that. You can see over here that this is an activating phosphate, and it gets removed here as part of the story in G1 regulation. Or is it G1 even? Yeah, we'll say. But we're particularly interested in these phosphates over here, and these phosphates are going to be removed. They're the inhibitory phosphates, and somebody has to remove them. We've emphasized a lot that phosphorylation is caused by kinases. I think it's the first time in our course that we learned that, well, when you want to remove those phosphates, you actually have a different enzyme, and the ones that remove them are called phosphatases. And so in humans, there are about 600 different kinases. And a kinase is an enzyme that stimulates a chemical reaction where a target molecule gets phosphorylated using ATP. So the products are the phosphorylated product and ADP. And for every kinase, there are phosphatases that catalyze the reverse reaction, removing that phosphate, and they're called phosphatases. And our phosphatase that does the job that we want to talk about is called CDC25 phosphatase. And the role for CDC25 phosphatase is it removes inhibitory phosphates from, and I'll just say from CDK, and I'll say CDK1 at the G2 to M transition. Very simple, when there is DNA present that's damaged, CDC25 is inhibited. So there's that little CDC25 enzyme that can catalyze that reaction, and when there's DNA damage, it turns out that CDC25 itself has an inhibitory phosphate added, and it is off due to DNA damage. So a really simple circuit. You gotta remove those inhibitory phosphates. CDC25 phosphates will do it. It's not gonna do it if there's DNA damage. Get the DNA damage fixed. CDC25 phosphates gets activated, removes the inhibitory phosphates, and we roll into mitosis. Jeremy and then Brooklyn.
So, do you have a question about this?
About reading it.
Yeah.
Yes, like what does that say?
At,
Removes inhibitory phosphates on CDK1 at the G2 to M transition.
So my question is from the previous two, because we were talking about like Aurora polo-like kinases.
Yep.
So when CDK is phosphorylated, is that what allows those, I assume they bind and form a complex on the CDK?
Aurora and Polo?
Yeah.
I'm pretty sure that they are just free, and,
They're free.
and it targets them and they get going and help it.
So CDK phosphorylates those
Mm-hmm.
to get going. Okay. So then the inhibitory proteins that are removed allow CDK and cyclin to complete the cell cycle. So are the two inhibitory proteins, when there's the three inhibitory proteins bound to the CDK, is that Aurora and Polo like activated, or is it when
No.
CDK is
Yeah,
dephosphorylated?
and when you say inhibitor, you've been saying inhibitory protein, but you mean inhibitory phosphate.
Excuse me.
So when the inhibitory phosphates are present, that is upstream of the Polo Aurora
Okay.
activation, because they get activated by CDK1 phosphorylating.
Yep.
So when there's DNA damage, CDC25 is phosphorylated and inhibited,
Got it.
and the inhibitory phosphates remain on CDK1. Polo and Aurora remain inactive because they haven't been phosphorylated by CDK1. And there are lots of kinases and phosphates and and phosphatases in this, but this is a simpler wiring diagram than the one over at START. Yes, Hannah.
So we talked about how there's like activating and inhibiting phosphates. So with START, that's the activating phosphates, and with this one here with the DNA checkpoint, that's the inhibiting like phosphates.
Not quite, but to start with where we are now, at the G to M transition, it's a DNA damage checkpoint
Yeah, and
that we're worried about. Well, just wait. And the DNA damage checkpoint is regulated by CDC25 phosphatase and removing inhibitory phosphates. Over at START, we're not going to talk about those phosphates very much because some play stories in activating and some repressing. It's complicated, and we can use just this story, right? It's over here. It's a nice little package to get a feeling for the role of phosphorylation in regulating CDKs. Make sense? What do we love about START? We love their cyclin-dependent kinases, so we have to make cyclin. Cyclin's a protein, so it has to have a gene, and that gene is regulated. Who's regulating it? Beta-catenin? Maybe. Maybe elk? Maybe. Maybe Mick? Maybe. Maybe June Foss? Maybe. Like that. See? That's START. Cyclin. We really want to study that. Then we're going to get around, we're going to loop around to the inhibitory proteins that bind at START. And we're just not going to talk much about the phosphates at START, just to mention that some activate it, some repress it, and there are just a big blizzard of stories from them, and would just, bury you in confusion easily. Here's an easy one. This one does have activating and inhibiting phosphates. We're just focusing on those two inhibitory phosphates that need to be removed. And who does it? and who regulates it. So it's a sweet package. Like I showed you in Intravial, this stuff is so significant. All eukaryotes have orthologs of CDC 25. That's sick. The yeast that causes bread to rise has an ortholog of human CDC 25. And they probably have 85% amino acid identity. Even though our last common ancestor, which we interpret must have had CDC 25, that last common ancestor would have lived 800 million years ago. Sick, right? It's sick. What modern biologists talk about loops in the history of life in a scale that human brains cannot possibly wrap our heads around. You know, that interpretation, the understanding of these relationships, and why we're talking about CDC 25, because it's profoundly important in our health. And just like the T-Rex, oh yeah, T-Rex would have been dealing with CDC 25 and DNA damage pretty much exactly the same way, no doubt, you know, like that. And of course, we'll never see that. T-Rex is dead, you guys, right? But yes, so let's have a concept chat. We're kind of running out of time, so we're just gonna have a concept chat and then quickly kind of get out of here. No yelling today, Dave. Dave gets a smiley face. We're just going to do this for another minute. We're going to run out of time. I did draw some purple up there because purple and black look really cool together. Hopefully that helps your learning. Does that help your learning a little bit? A little bit, though. Purple and purple and black. Oh yeah, is that your comment? All right then. Let's talk to Sophia and Brooklyn both. That's a dodge. I call that a dodge.
Okay.
Yeah, what is your question? What is one important thing we talked about today? Oh, what thing? Sorry, go Brooklyn, go.
We talked about Kit, and how cool that is and how it's mutated in gastrointestinal
Yep.
tumors, and how it's a tyrosine kinase, and how it contributes possibly even if it's not mutated. That's something cool to discuss.
Yeah, I mean, we were just kind of speculating on what might, that's, I think, kind of future looking about what the explanation for those percentages would be. But yeah, Kit is epic story, right? People are really concerned about those polyps in intestines, right? And you know, they measure them. What do they measure? Well, mitotic index, polyp size. There are some other features they add more and more. Some are morphological, so they talk about some different types of polyps they can see. Similar to on the surface of our bodies, the skin moles we see, they categorize those in some different ways. But now they also, when they remove some polyps, they'll do some molecular gene expression analysis, gene expression and mutation analysis. Yes. So Zofia, what's another thing that you guys talked about? Kid and something else.
Yeah, well, a lot about the CDK and how growth factors can stimulate or inhibit cell death, and how they need to have CDK. Talked a lot about like the G2 to F phase and like that whole transition.
Yeah. Well, that first thing is great because again, that's why the next chapter in our book is about apoptosis and programmed cell death. It's because lots of growth factors will regulate that as well, and as a result, there are some genes in those programmed cell death pathways that get mutated in cancer. So we get cancers that are growing too fast, too much stimulation of the cell cycle, and no programmed cell death check. You know, the cells are just growing, going, no, I think we're cool. This is good. We're doing good. And then, Joe, what's the one other thing we talked about?
We talked about the animal embryos and how they only have an M phase and an S phase, and they don't have a G1 or G2. But they, inside with the animal embryos, they're given like all the building blocks they need. And we also talked about the meiotic index. And I just had a question with that. With the meiotic index, how we were saying it's like low in normal skin and like underneath the scab or the lady with the skin cancer, it's higher. The meiotic index, how does it respond like a lady with the skin cancer, like when she's going through treatment? Does it get lower as she's going through treatment and the cancer cells are dying? Does the index go lower or does it stay like the same?
Yeah, so what about somebody who has cancer who's going through some chemotherapy?
Yeah.
And my answer would be, I don't know. Like that? Yeah, well, I don't know. But then there would be things that you would say that would be some examples, and it would be a variety of things that how the mitotic index might be affected. In some cases it wouldn't be affected, in other cases it would be affected like that. Yep, yeah, and I think we're out of time, so we just have to stop here. And so the sign-up sheet is somewhere, and so sign up if you didn't see it. Come on Thursday. We'll hope that Riley is back in action and with us on Thursday. And we'll talk about cell cycle, but we'll really, I'm sure.
Just a follow-up to his question.
Yes.
You said under the normal scab, there's a high